All right, let's dive right into this explainer. Today, we're exploring the fascinating, highly structured world of the plasma membrane. We're going to take a journey to the very edge of the cell, where one of nature's most incredible balancing acts is taking place, literally every single second. Think about a highly exclusive VIP club. Inside, everything is perfectly controlled, but outside, it's absolute chaos. So how do the bouncers know exactly who to let in and who to keep out? How does the cell communicate with its environment without losing its own internal identity? Well, that is the job of the plasma membrane, and today we're going to decode exactly how it pulls off this incredible feat. Here is our club map for today's explainer. We'll start with the anatomy of the boundary, move on to the fluid mosaic model, check out passive transport, which is our free entry, and finally look at active transport, or VIP access. Section 1. Anatomy of the boundary. Building the VIP club. It's crazy to think it took almost 50 years to go from discovering the basic two-layer lipid fence to actually understanding the dynamic, bustling model we use today. Back in 1925, scientists Gorder and Grendel were looking at mammalian red blood cells. They extracted the lipids and realized, hey, these form a two-molecular thick layer around the cell. But it wasn't until 1972 that Singer and Nicolson finally proposed the fluid mosaic model, which absolutely revolutionized our understanding of this boundary. So, what does this basic fence look like? Well, the membrane is mostly made of phospholipids arranged in a bilayer, a double layer. Picture these phospholipids like little tadpoles. They've got polar heads that absolutely love the watery environment on the inside and outside of the cell, so they face outward. But their tails? Those are hydrophobic. They fear water, so they naturally tuck themselves away on the inside of the bilayer. It's an elegant design that creates an immediate, highly effective barrier against the outside world. Now, if you look closely here, notice how the densely packed, water-loving heads on the top and bottom basically sandwich those water-fearing tails right in the middle. It effectively separates the inside from the outside. Under a high magnification microscope, it almost looks like a tiny railroad track. But as you'll see in a moment, this structure is far from just a static brick wall. Section 2. The fluid mosaic model, a bustling, dynamic boundary. Right, so this boundary isn't just a rigid shell. It is alive, moving, and highly intelligent. Singer and Nicholson's fluid mosaic model tells us that the lipid bilayer has a quasi-fluid nature. What does that mean? It means proteins can actually move laterally within it. It's like a crowded dance floor. This ability to flex and adapt is measured as its fluidity, and it is absolutely crucial for things like cell growth, division, and secretion. Now, you might assume a lipid bilayer is mostly just, well, fat. But check out this data. In a human erythrocyte, a red blood cell, the membrane is actually 52% protein and only 40% lipid. That means in our cellular VIP club, by weight, there are literally more bouncers than there are sections of the fence. So let's meet these bouncers. The proteins in the membrane are classified by how easily they can be extracted. First, we have peripheral proteins. These guys just hang out loosely on the surface of the membrane. Then we have integral proteins. These are partially or even totally buried deep within the lipid bilayer, acting as structural gateways. Oh, and we can't forget cholesterol. In most eukaryotes, cholesterol is wedged into the membrane to provide vital structural rigidity. It basically holds the whole club together. Section 3. Passive transport. General admission, no energy required. Let's see how this boundary actually works when molecules try to enter the club without a VIP pass. Welcome to the general admission line. This is passive transport, where molecules naturally flow from an area of high concentration to an area of lower concentration. And the best part? The cell doesn't spend a single ounce of energy. If it's a neutral solute moving across, we call it simple diffusion. If it's water moving down its concentration gradient, that's osmosis. Essentially, it's free entry for the right kind of guests. But of course, there's a catch. Because those lipid tails tucked inside the membrane are nonpolar, polar molecules get rejected right at the door. No exceptions. They just can't pass through that water-fearing middle layer on their own. They're going to need a designated door. So, when a polar molecule approaches, instead of just bouncing off, a special carrier or channel protein steps in as a gateway. We call this facilitated movement. A great example? Plant and animal cells use these critical channel proteins called aquaporins just to transport water efficiently. The guests still get in for free, meaning no ATP energy is spent by the cell, but they have to use these specific VIP doors instead of just walking right through the walls. Which brings us to Section 4, Active Transport, the VIP entrance. What happens when a molecule needs to sneak in against the natural flow of the crowd? Well, there's a massive difference here. Passive transport is easy. It's like floating downstream. But active transport, that's moving against the concentration gradient, going from a lower concentration to a higher one. To pull this off, the cell has to pay a VIP cover charge, and that currency is ATP energy. Because it's an energy-dependent process, the cell actually utilizes those ATP molecules. When something needs to go against the gradient, specialized membrane pumps literally force the molecules across the boundary. The absolute poster child for this is the sodium-potassium pump. It works tirelessly, especially in our nerve cells, to actively transport ions across the membrane, maintaining that precise internal balance the cell absolutely needs to fire signals and, well, survive. Bringing it all together, the plasma membrane being selectively permeable restricts molecular movement and maintains cell composition. That selective permeability is the absolute core of cellular survival. By perfectly balancing the lipid barrier, the fluid mosaic of proteins, and that intricate dance of active and passive transport, the plasma membrane acts as the ultimate intelligent border security. It decides exactly what stays out, what comes in, and what gets kicked out. It is an incredibly elegant system, right? But it does leave us with a fascinating final thought. If this cellular gatekeeper is so incredibly complex, so highly selective, and so secure, how exactly do invading viruses, or on the flip side, targeted life-saving medicines, manage to trick those protein bouncers and sneak inside? I'm going to leave you with that provocative thought to ponder. Thank you so much for joining this explainer on the dynamic world of the plasma membrane. Keep asking questions and keep exploring.
Thank you for watching. See you in the next one. Stay tuned.